Hello, everyone, and welcome back to Day 2 of Cell and Gene Therapy 2025. My name is George Hammond, Senior Conference Producer at Technology Networks, and I'm pleased to welcome our opening presenter for today, Dr. Neal Flomenberg, presenting his talk, Genetically Unmodified Cytotoxic T lymphocyte therapy for viral infections, their sequelae, and cancer. Neal graduated from the Sidney Kimmel Medical College of Thomas Jefferson University in 1976. He began his medical oncology career at the Memorial Sloan Kettering Cancer Center in 1981 in the bone marrow transplant program, working as both a clinician and laboratory investigator in the areas of T-cell physiology, histocompatibility, immunologic redevelopment, and improving the outcome of patients receiving mismatched blood or marrow transplants. In 2022, he joined Tevogen Bio as a chief science officer. Tevogen is focused on using genetically unmodified CTLs to treat viral infections, the consequences of viral infections, viral-induced cancers, and sporadic cancers. Please join me in welcoming Dr. Flomenberg. And following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And now, without further ado, I'd like to hand over to Neal to begin his presentation.
Good morning. Pleasure to be here. Let's dive right in. So cytotoxic T lymphocytes, or CTLs, are nature's defense against viral infections and more beyond that. They've been used for a while. They have been used in the aftermath of bone marrow transplant, when people's immune systems were pretty much devastated, and viruses like Epstein-Barr virus and cytomegalovirus reactivated, and have been proved to be safe and reasonably effective. That said, one of the things that we do a little differently, that I'll be speaking with you about, is that for any treatment that you give a patient, you need precision about what you were targeting and what you were giving to the patient. Similarly, like other treatments, the dosing must be consistent patient to patient to patient. Given that viruses do change and mutate, when a new variant arises, you need to be able to rapidly assess the impact of the changes in the pathogen on the treatment that you're offering the patients. Ideally, as soon as it's sequenced, you'd like to know. And then beyond the issues of safety and efficacy, you'd like treatment to be rapidly available and conveniently available to the patients. So as I mentioned, a lot of the background in this field came from bone marrow transplant patients with Epstein-Barr virus reactivation, post-transplant lymphoproliferative disorder, as it's known, or cytomegalovirus reactivation. There are hundreds of patients that have been treated over decades. There are hundreds of patients that are reported in the literature, and that's probably just a fraction of the total number that have been treated. The therapy has generally been extremely safe. It's not been associated with the cytokine release syndrome or neurotoxicity events, as has been seen after CAR T therapy. It's been an efficacious treatment. The specific percentages of patients who respond vary somewhat with the virus. They vary with the post-transplant characteristics of the patient. Is the patient still on immunosuppressive therapy? How far out after transplant is the patient? And other clinical variables, but there is a good record of efficacy as well. And people who have been in the area have suggested that somewhere around 20 of the most common HLA alleles would provide treatment opportunities that would allow us to cover most of the population. So I'm going to take a moment, since I believe that it's a somewhat heterogeneous audience that's watching this, to talk about the ways cytotoxic T lymphocytes work. And I'll be focused primarily on viral infections, though the principles are similar for other sorts of things. And as the title of the cell indicates, CTLs actually focus on a cell's trash, and that's what they utilize to recognize whether the cell is normal, infected, what have you. And shown on the left is an example of a viral infected cell in red. In the HLA class 1 molecule is a small peptide fragment, which is the antigen that the T cell recognizes. This is recognized by the T cell receptor. And the CD8 molecule, shown here as well, is a co-stimulatory molecule which recognizes some of the non-variable portions of HLA. Now, on the right, we have a reproduction of a figure that was in Pam Bjorkman's paper when the first HLA molecule was crystallized. People understood at the time that antigen, peptide in this case, and HLA were seen together, but they really had no good understanding as to how that happened. This is looking down on the molecule, the HLA molecule, as a T cell would see it. And in the left, you see the HLA molecule by itself. And on the right, you see a peptide in red, which was co-crystallized along with the molecule. And it produced a major aha moment for cellular immunologists everywhere. Now it became clear as to how the processed proteins and the other trash in the cell, were seen together in HLA molecules. It essentially was holding it up for the T cell receptor to see. And you can, imagine that both the blue portions that are most projected away from the cell and the red portion are both seen together by the T cell receptor. Now, if I can sort of pause for a moment and sort of try and deal with some perplexity of the non-cellular immunologist in terms of trash. When I teach this material, I always use a common example. Down the block from you is a little old man and woman who keep their house absolutely immaculate. They happen to be vegans, wouldn't touch anything dairy or any meat products in terms of what they eat, and they're teetotalers and would never touch a drop of alcohol. You walk by their house one day, and there are no strange cars around. There's nothing, that's broken or damaged or looks awry on the outside of the house. Everything looks completely normal. But you happen to notice in the trash can that there are a whole bunch of beer bottles and pizza boxes with remnants of pepperoni pizza. Now, given what these two people eat, you know that something is different in the house. You don't know whether their grandkids are visiting because college is on spring break, or whether they're being held hostage by a group of biker gangers. It could be anything in between. In the body, there are no benign visitors inside of cells. They're all problematic. And this sort of illustrates that just by looking at the trash, even when everything on the surface looks normal, the immune system can recognize that it's things are awry. HLA varies throughout the population. It's obviously what we use to match patients for organ transplants. Prior to having modern medicine, this helped keep us safe. If there were some HLAs that were better with a particular infection, and others happen to be worse, at least the people with the good HLAs for that infection managed to sneak through and deal with the process okay, so that the species as a whole managed to survive.
When a cell gets infected, it produces viral proteins, which are broken into peptides. Human leukocyte antigen molecules, or HLA, bind these peptides and display them on the cell surface. Cytotoxic T cell receptors recognize foreign viral peptides displayed by HLA. Each T cell expresses a single, specific receptor type. However, receptors are highly variable to detect all antigenic peptides. During infection, T cells recognizing the specific peptides activate and multiply. Cytotoxic T cells kill infected cells, displaying viral peptides, allowing the body to replace them with healthy cells.
All right, so I apologize in that this particular way to play the slides didn't give me any time to talk before the slide went live itself. So what I'm trying to illustrate in the next couple slides is how our production methods of producing large numbers of these T cells reproduce an actual viral infection, the parallels between them. An infected cell doesn't only have a single HLA, they have plenty. And the T cells similarly have lots of T cell receptors, though they rearrange their T cell receptors the same way B cells do to produce a single T cell receptor or a single antibody from each immune cell. So we're going to move on now and look at how we use this process as to how T cells work when confronted with an actual infection to grow our T cells in the laboratory.
The exact T cell platform mimics nature's process, providing patients with effective immunity more quickly than their bodies can generate on their own. During manufacture, healthy cells are coated with infection-specific peptides. The peptides bind to HLA and activate T cells, prompting them to divide as they would in response to an infected cell. We do this 3 times during manufacture. We get at least 600-fold increase in the number of T cells during this process. After the 3rd stimulation, the cells are harvested, adjusted to the required concentration, and frozen. Our cells are highly potent and specific, with hundreds of doses produced per batch. Frozen cells remain viable for years, with stability testing showing Tevogen 489 meets release criteria even after 3.5 years.
And Tevogen 489 happens to be our COVID product, which I'm going to sort of walk you through in terms of the trials that we've done and some other aspects in terms of its efficacy and stability. Now, since we're talking about COVID, this is the COVID viral genome. It's got a large open reading frame 1AB, and there are other smaller open reading frames that are given these numbers. They're all ORF followed by the number. The structural proteins are given letter designations. Spike is here, S. And I would comment that all of the structural proteins put together represent about 20% of the viral genome. Spike is 13% of the total. So in terms of the vaccination approach that's been used towards spike, it leaves most of the rest of the genome and its potential targets behind. The non-structural proteins, again, are about 80% of the amino acids. Now, one of the things that we've done at Tevogen is to create a database where we literally have every single sequence that's been reported to NCBI. And the database, ranges in the millions. By far, spike has the highest mutation rate of all of the proteins within the genome, which is a small headache or a big headache in terms of immunity post-vaccination. So, our platform, we're trying to use nature's approach and bring CTL therapy to vulnerable subgroups faster and more robustly than they can do so on their own. Our goal is not to treat healthy 30-year-olds. Our goal is to treat immunocompromised individuals, people who don't make a prompt enough immune response because of age or other debilities, and that sort of thing. Now, obviously, the definition about who's vulnerable depends on the virus that you're talking about. COVID for a healthy 30-year-old now is probably a bad cold. The Ebola virus kills about 50% of the people that it infects. So obviously the definition would vary depending on what the target is. Now the CTLs, as we've discussed, typically are directed toward short viral targets. And we select a number of these multiple targets spread across the genome rather than limiting ourselves to spike and worrying about the consequences of mutation. And I will show you some data later on about the durability of our product. For all virus products, as a start for trials, 6-HLA restrictions can cover about 2/3 of the population. Our strategy to move beyond this is to try and balance two issues: one, maximizing patient coverage, and two, not leaving any racial and ethnic group behind. The HLAs that are most common do vary across racial and ethnic groups. HLA A0201 is the most common in the world and is in the top 10 in all racial and ethnic groups. And that was the one we focused on in our phase 1 study. Now, the T cells are frozen. They're available off the shelf. And when you're not doing dose escalation studies where you've got to cover a wide range, they can be delivered as less than a 20 cc, infusion. Tevogen 489, which is directed against COVID, is the first product that's been developed from our Exact T cell platform. We start with computer prediction as to which peptides are going to work, and then we go on and confirm those through lab work using an approach that's similar but on a smaller scale than our manufacturing approach. Now, I talked about quantitation, and this is something that we feel relatively strongly about. So how do we quantitate, the cells? We use something referred to as tetramers. Tetramers are small beads which have 4 HLA molecules of a particular specificity. The beads are then coated with target peptides during an incubation, and they have a fluorochrome, and so they can be used for flow cytometry. So if we look at this figure backwards, after the third and final stimulation in terms of this batch, and I should indicate CD8 is on the x-axis and tetramer staining is on the y-axis, 76% of the cells stained with the tetramers that we were using. And less than 5% of the cells were not CD8. 95% of the cells were CD8 positive. Now, if we go backwards, after the first stimulation, you can probably see, and I don't know how well the dots show up on your screens, a small population that's at 2.3%. If we went back before the first stimulation, you wouldn't see anything at all. So, we've gone from something that's less than 1% up to 76% in this particular batch. And we have a number of process improvement steps that are underway that we're cautiously optimistic should get us up to about 90% tetramer positivity. At the present time, we would argue that dosing should be based on tetramer positive cells rather than total cells in the product, just like other drugs have the active ingredient and then have inactive ingredients that are used to make the tablet that patients take. Once we get up to 90%, it probably is immaterial in terms of the difference between active and total. Now, we get large expansion per batch, and in terms of walking through that, for our phase 1 study, we knew that to cover the 4 dose levels, we wanted somewhere in the area of 8 to 9 million cells. So we started with 8 to 9 trillion cells. So we started with 3 trillion prior to stimulation 1, and by the time we were done, we had a little over 9. So we had a little bit more than a 3-fold expansion. That said, the CD8 percentage went from 39% at the beginning to 95%. So it wasn't all the cells, again, that were growing. There was another 2-fold expansion within the CD8 population. And both of these are underestimated, again, so that we're not, inflating the numbers. With regard to tetramer staining, it went from undetectable to 76%. So for the purpose of calculating the expansion, we used 0.76, which gave us a nice, 100-fold number. In actuality, if you look at memory T cells after a viral infection, which is, what our starting, point is, they're usually 1 in 1,000. So 76% is actually quite a generous number. And if you multiply these 3 numbers together, this is where we come up with a 600-fold expansion in terms of the tetramer positive cells or the specific cells that happen to recognize the COVID virus. Now, that's great. They bind to the peptides. Are they functional? So the classic way of looking at CTLs is in a chromium release assay, where you have both positive and negative targets. And what we used here was an A2 positive cell line for these A2 positive CTLs. And it's shown in 2 conditions. At the top, after being pre-incubated with peptides, so that the COVID peptides were all there. There's an equilibrium between the ones presented by the cell and the ones that you present exogenously, and they get coded. And the exact same cell line, which was not coded. And you can see that there's a huge difference. Down to 3 to 1, we had more than 80% of the targets killed in a 4-hour assay. When you went from 3 to 1 to 1 to 1, it started to fall off a little bit. That's still quite good based on how CTLs work and how many are usually required to kill a cell. So from the point of view of what's going on in the body, this basically means, infected cells would be killed off, where uninfected cells are left alone. Now, there are other people, working in the area, and, I will say that in contrast to us, where we're focused on a limited number of peptides, a lot of them will use either intact proteins or in many cases, peptide libraries that may have thousands of peptides in there. As one of the ways to get around the number of peptides needed in a peptide library, one of the things is to make them longer. And that makes them bind a little better to HLA class 2 molecules rather than class 1, and they, stimulate more CD4 T cells. We'll show you our data. There are other people that have, tried products that instead of being mostly CD8, were mostly CD4, and those trials were not successful against COVID. So to try and summarize a little bit about what's different, the entire viral genome's targeted. The targets are far more durable. Now, we started with this process in 2020, and what is shown in the table on the right-hand side are the percentages of our targets in our COVID database, going through some of the more recent FLIRT variants. And you can see that the numbers are quite high, in the high 90% range. In contrast, in terms of monoclonals, there's a list of a lot of them here. Mutation frequently renders them ineffective, and their EUAs were revoked. The T cell targets, if you don't mutate them right in the middle of the peptide, you don't really have, for the most part, an effect. For B cell targets, things that are remote from the target, the binding site, can alter the way the protein folds, its shape, and they can interfere with binding even though it's not directly at the site. We're the only company generating CTLs that are restricted by one and only one HLA. That allows for consistent dosing patient to patient. So everybody who's HLA-A2 positive gets the same dose. When you make them, against a peptide library, you generate them against two HLA-As, two HLA-Bs, two HLA-Cs, and then the dosing toward for a particular patient is variable. And again, because we know the sequences of these targets, if there's a new variant, as soon as it's sequenced, we can assess the impact. Now, we've taken this through a study design. These were ambulatory patients with newly diagnosed COVID-19 who were at high risk for a poor course and a poor outcome due to one or more of the CDC's defined high-risk criteria. The FDA required that we treat everyone within 4 days. Patients who were A0201 received the CTLs. People who were negative were in an observational arm. The interventional patients were treated with TVGN 489 and observed in the hospital for 4 days. And this was an FDA requirement, not something we wanted to do. The FDA was concerned about cytokine release syndrome from CAR T cells. And there was a phenomenon in COVID that had been named early in the pandemic, cytokine release syndrome, with a completely different pathophysiology. But the combination made them a little uncomfortable. We treated 4 dose levels over a 30-fold range, and these were based on the doses used in the bone marrow transplant patients that I discussed before. After the 4 days in the hospital, they were discharged and followed for another 10 days, so for 2 weeks total, and then had follow-up at 1, 2, 3, and 6 months. And dose-limiting toxicities were monitored during the first 14-day period. The primary endpoints: did they have any infusion reaction to getting the product? Did they have cytokine release syndrome as seen after CAR T? Did they have neurotoxicity as seen after CAR T? Did they have transfusion-related graft versus host disease? And did they have any grade 4 or higher adverse events thought related to the CTLs? Secondary endpoints in the interventional arm were viral load reduction by follow-up nasal swabs, persistence of the cells for up to 6 months, the 6 months that we were following them, and assessing whether the humoral or cellular immunity to COVID was in any way disrupted by giving them the exogenous product. And then the exploratory endpoint was to look at symptoms and see if we had symptomatic control. So here is the interventional arm and the observational arm. The age is pretty comparable. The ethnicity is fairly similar. The gender distribution is fairly similar. There were more immunocompromised patients in the interventional arm. So these were more people on cancer therapy or with autoimmune disease and that sort of thing. There was a slight increase in the number of comorbid conditions. This was not huge, but it, again, was making the interventional arm look a little tougher. And in terms of baseline antibody status, there were a higher number of people that were either unvaccinated or because of chemotherapy or other drugs had no response to vaccine that they had gotten. We started with the Delta variant, which was one of the more aggressive COVID variants. And I would just indicate that, that more aggressive variant actually got our lowest dose level. And we finished the study through a variety of the Omicron variants. You can see the issues that the patients had, some cases cancer, obesity being common, hypertension. This patient had asthma and also some problems with sleep apnea. There are a variety in all the patient groups. When it came to side effects, it's relatively easy to say there were none in terms of the things that we had defined. Our Institutional Data Safety Committee and our external medical monitor reviewed the results and deemed the therapy well tolerated at every dose level. If you look at the exploratory endpoint of when the patient started to feel better, I walked into the room of the patient who was number one. This was when I was still at Thomas Jefferson University, where the study was run. And the morning of the third day, he had gotten the cells in the evening on day 0. In the morning of the third day, he said to me, "Doc, I don't know what happened, but last night I started to feel so much better. I feel like I'm pretty much back to normal now." Didn't really know what to make of it, but that became a consistent pattern that we saw patient to patient to patient. In the observational group, there was much more variation. Now, this patient is particularly interesting. This one here is the biggest outlier. He was the husband of one of the second patient in the interventional arm. He had the Delta variant. She was in the hospital for 4 days as required, went home, and the next day, of course, she scrubbed the kitchen floor. What else would you do? He, unfortunately, in the observational arm, because he didn't match the cells, landed in the hospital, went to the ICU, and then did a stint in rehab. Obviously, it's pretty likely that the two of them had the same variant of the virus. We did not see any COVID reinfection and no evidence of long COVID in any of the treated patients for the 6-month study follow-up. And by the time we got to the 4th dose level, even the FDA was convinced, and they approved the cells for outpatient treatment by the end of the study. Now, that patient who told me he felt better when I went in on day 3, didn't know what to say, placebo effect or what, but we did his viral load on day 4, and to my amazement, and I still get choked up when I think about it, his PCR was negative. I didn't say anything to anybody until we confirmed it again on day 7. Now, we didn't see everybody going in day 4, but everybody got rid of 99 to 100% of the COVID by PCR on day 14, by day 14. 2 immunosuppressed patients got rid of 99%. The other 10 got rid of 100% and were considered, fully negative. Some of them had variable courses. The second patient told us she had a clogged maxillary sinus, and her viral load actually blipped up a little bit right after she told us that her sinus was now open and draining. Probably there was a reservoir of COVID in there, which, when opened, pushed the levels a little bit. Now, we wanted to see how long the cells lasted, and we worked with Adaptive, which is a company that does T-cell receptor sequencing. We gave them several different 80201 products, And to our surprise, though they recognized the same peptides in very similar proportions, they were actually quite different in terms of their T cell receptor sequences. So what Adaptive did for us was to look at sequences which were present in the product, but missing from the patient. And you see that they blip up, quite rapidly after the cells are infused. But to our surprise, they still lasted 6 months in the patients. And that was not something we expected. In the bone marrow transplant experience, cells were gone much more quickly, although the approach that they used was quite distinct. Now, if we go back to the left-hand side and, the cells are persistent, there was a recent publication that suggested that the clearance of COVID in immunocompromised patients was 72 days. And we have the first author and the reference here, if anyone wants to look at it. So obviously being rid of 99 to 100% by 14 days when compared to this, looks pretty significant. So all treatment patients returned to their baseline level of health within 14 days of treatment. No patient developed long COVID, at least up through this time of the 6-month follow-up. No patient in the interventional arm has had either a recurrence or rebound of COVID after CTL treatment. No impairment in the patient's own cellular or humoral immunity occurred from the CTL administration. So, when somebody is immunized, they make spike antibody. When they get an actual infection, they make antibodies to some of the other structural proteins. We did not interfere with that at all. And one of the more interesting events is that 2 of the cancer patients on the study went on to stem cell transplant right after the follow-up period ended or within a month. Now, obviously, if we hadn't gotten rid of the vast bulk of COVID, you would have worried that it might have reactivated after we blew away the patient's immune system as part of their transplant procedure. Bone marrow transplant, stem cell transplant is about as immunodestructive as you get. Neither of them had any reactivation or any problem with COVID, despite having a bone marrow transplant within a month of getting CTLs. So in terms of where we'd like to go, we're going to look at humoral immune suppressed patients, and we're going to focus on cancer patients with B-cell malignancies and getting therapies that further aggravate the B-cell suppression. We're going to look at cellularly immune suppressed patients, transplant patients, or those on immune suppressive drugs. We're going to look at elderly, debilitated, problematic comorbidities. And the common theme between these is that they're patients who can't respond robustly enough or in a timely enough fashion to the virus. The other point that I would make, with regard to the B-cell malignancies and suppression, is that if you put a large delay, a large hiatus, a large gap in somebody's cancer treatment, even if that treatment was likely to be curative, you really compromise the chances that that's going to happen, a great deal. So treating these patients so that they can quickly get back to treatment, and we've shown that people could even move on to stem cell transplant relatively quickly, is certainly in their best interest. Now, there is, another rationale for treating some of the immune compromised patients, and these are 4 papers from relatively distinguished journals, and they talk about evolution in immunocompromised hosts of the COVID virus and have suggested that disproportionately, this is where new variants come from. So there may be a benefit, really, to a public health benefit to treating these patients to help avoid the emergence of new variants in the future as well. What else are we going to try and do? There is a growing body of data which suggests the existence of a COVID reservoir in long COVID patients. Some of the other thoughts about this syndrome is that it's the reactivation of some of the herpes family viruses shown here because of immune dysfunction in the consequence of COVID infection. So as I indicated, thus far, none of the patients treated developed long COVID. I agree that it's a modest sample. And this suggests to us that rapid and vigorous clearance of the virus may prevent long COVID by preventing a reservoir from being established and or immune dysfunction occurring due to a more aggressive COVID source. Other targets, myalgic encephalomyelitis, also known as chronic fatigue syndrome, is something that's been known after EBV for years. It's similar in many respects to long COVID. It's got a lower frequency. 90% of the human race gets infected with EBV, and this is a small fraction of the total population. So similar, but less frequent. There's growing evidence that EBV is the trigger for multiple sclerosis. In a small percentage of EBV infected individuals can this be targeted. Chronic hepatitis B has no curative treatment. There are things that can suppress it, but you got to take it for a lifetime. It causes liver failure and liver cancer. Human papillomavirus causes cervical cancer and other cancers. EBV causes lymphomas. Now, sporadic non-viral induced cancers are being treated with immunotherapy in some cases. As we learn more about the targets, we can go after them as well with CTLs. There is a new and emerging field as to what is referred to as the cryptic peptidome, which may provide targets to go after in malignancies. These are things where the splice site between introns and exons is wrong, or the frame in which the RNA is read gets shifted and it's a completely different sequence, or other sorts of things. I don't want to go through all of them. These are not things that we have been rendered well tolerant to. And you can imagine a scenario where when the cell is sick, either with a virus or cancer, this is going to become more problematic, higher frequency than it would be under normal circumstances. And what we're hopeful is that somebody can see a doctor by day, have their nose swabbed, get a rapid HLA typing done, which is what we did for all the patients. We had them back in 4 hours, get treated that afternoon, and hopefully sleep in their own bed that night, which is, one of the things that'll make the treatment well tolerated and well accepted. These are my colleagues. This is Dr. Sadi, our CEO, who is extremely passionate about healthcare equity and affordability. This is me some years back when I was a little younger and a little less gray. Sadiq Khan is our commercial officer. Kurti Desai takes care of the finances. Steve Chen is our chief technical officer. Mittal Mehta is our chief information officer and head of Tevogen AI. Dolores, or Lori Grosso, is our clinical development lead. She and I have been partners for over 25 years, both at Jefferson and Tevogen. And Ali O'Connor is our senior scientist and leads our current lab efforts. So I thank you for listening. I'd be happy to take what questions that time allows.
Thank you so much, Neal, for that fantastic talk to open day 2 of Cell and Gene Therapy 2025. We really do appreciate you sharing your expertise with us today. So we're likely going to have lots of questions submitted during the Q&A, and I can see that we have received some fantastic ones already. So I'll move straight onto the Q&A now. So Neal, your first question is, are there any ways that you can anticipate to increase the cell yield further?
So we are actively working on that. In the early parts of my career, I grew clones of T cells where you limiting diluted the starting point so that roughly 1 in 3 wells would grow, and typically from a single progenitor. And we were able to get millions of cells from those. Right now, The limiting thing is not the lymphocytes, but the antigen-presenting cells that we use to stimulate them. We have ideas as to how, when we get the purity up very high, we might be able to continue stimulation without antigen-presenting cells. So I think that the answer is yes. I would say that, we don't have a firm dose established because every dose was effective. If we can use the lowest dose that I've shown here, we are going to get many, many hundreds of doses from one batch, which should help to make the product highly affordable and accessible as well.
That's excellent and a fantastic answer. Thank you so much, Neal. Now, we have a wonderful question from Heather in the audience, which is, how do you test the safety endpoints that you mentioned during your presentation? So the cytokine release, neurotoxicity, etc.
Well, I mean, the patients were observed in the hospital for 4 days. In the bone marrow transplant field, we look at patients for whether they have fevers, shortness of breath, various, kinds of symptoms during the infusion. For those products, you're giving hundreds of ml in many cases. Where we hope to be with this is at 20. We gave some people about 150 because we had to really produce a very dilute product to be able to cover the 30-fold, dosing range. Neurotoxicity, there are scales, that are used for COVID patients. I'm sorry, for CAR T cell patients, pardon me. Cytokine release syndrome, there are defined criteria. They just didn't have any of that stuff. And they were monitored in the hospital. Beyond me going around, there were interns, residents, other physicians, of a more senior level, lots of doctors, nurses, so lots of people observing the patients.
Excellent. Another fantastic answer. I believe we have time for just one or two more questions today before we close out this session. So are there any limitations of the platform that you discussed in regard to mutations in virus target antigen?
Yeah, so that's a good question. So COVID's a new virus. And, though it has mutated a lot and variants have come up, they've sort of been manageable. If you contrast it with other viruses that have been part of the human race since the human race started, there's been lots more evolutionary time. In terms of some of these other, viruses, it's a little more challenging. It's doable, don't get me wrong, but the longer period, and it's a far longer period that the viruses have had to mutate, does make for some challenges. One of the thoughts, with regard to a highly mutated virus is to take a large number of targets and basically give enough cells so that if you cover half of the targets, you've got enough CTLs to be sure that you're giving an effective dose. And that's one of the things. There are other ideas as well.
That's excellent. And we have time for just one final quick question today, which is, if this therapy has this much potential, why hasn't it been used to a greater extent before now?
Yeah, it's a good question. And the answer is, it's not a one-size-fits-all treatment. And that's typically what most people want. One drug that you can give across the board, not that you've got to make an HLA-A0201 product and an HLA-0101 product and an HLA-1101 product and an HLA-2401 product for all the patients, and do all the homework that's required in terms of the peptides, because they are different from one HLA to the next, to the next, to the next. So I think it's the fact that it has not been a one-size-fits-all product that has held people back. And I, give credit to my Tevogen colleagues that that's not stopped us, and that we're willing to take that on and deal with it and move forward. So best answer I can provide.
That's wonderful. Thank you so much, Dr. Flomenberg. Unfortunately, that is all that we have time for today.
My pleasure. Thank you.
Thank you so much, and once again, for such a fascinating session. Now, just a quick reminder to our audience that any questions that we didn't have time to get to will be sent to the speaker after the event. So that just leaves me to say a massive thank you once again, Dr. Flomenberg, and to everyone in the audience. We'll be back soon with the next talk.